Good afternoon, everyone. So hopefully I won't put you to sleep this late in the afternoon. While I tell you about evolving blight diseases. So this title was given to me by Dirk, I guess to keep in touch with the evolve. And if you talk about blight diseases, of course the most famous or rather infamous one is late blight, which was the causal agent of the Irish famine. Now, this organism is not a fungus, it is an oomycete. It's called Phytophthora infestans. Now, Phytophthora translates to plant destroyer. Now, as you can imagine, if this caused a famine, it is really, really destructive. So, in 1845, the population of Ireland was about 8.4 million. By 1900, it had reduced to 4.4 million. Now, at least not all of these people died of hunger, only about 1 million. A lot of them emigrated and other causes. But yes, so this pathogen can be really devastating. But thanks to innovation and research, these days we don't have that much of a problem with it, especially in South Africa. But every now and again, epidemics happen if the conditions are conducive. So very, very characteristic of this disease is this light green halo around the lesion. Now, we've done a little study now for Potato South Africa, where we did a survey of late blight in the country, and despite lots of problems trying to get samples, we did manage to get a population together, and good news, so far we still only have the A1 mating type. And this is important for us, as this pathogen needs two mating types, A1 and A2, in order to reproduce sexually. So currently the population in South Africa can only reproduce asexually, which means it doesn't evolve quite as fast, which is good news for us. We also looked at some of the fungicide resistance, or if they are still susceptible to the fungicides that we are currently using. And while we are still busy with these tests, good news so far, everything that we've tested are still susceptible to these active ingredients. So far so good. Now, if you look at the late blight symptoms, very characteristic if you have conducive conditions is the sporulation on the underside of the leaf. And if you see that, then you know you have a lot of inoculum in the field and you have to really respond very quickly to do something about it. In South Africa, however, if the conditions are not conducive for the pathogen anymore, the lesion will actually dry out very quickly and the leaves will shrivel up. Now, if you look at some of the control options for late blight, so resistant cultivars, of course, is the best way to go because then you can use less fungicides, but then good field drainage and proper plant spacing is also very important. Then proper sanitation, if you have any coal piles, do not leave them lying around because then there will be more inoculum in the field. The irrigation can be monitored if it's not raining, and if you can ensure that the leaves are dry in the evening, then the infection will not be able to happen either. Excessive fertilization will also be conducive for the disease, but then luckily the contact fungicides are still effective and some systemics as well. So moving on to the next blight disease is the early blight, which is a much more of a problem for us in South Africa. Very characteristic is the concentric circles inside the lesions. Now, the name might say early blight, but it doesn't mean it comes in early in the season. It actually comes in later in the season most often. And then it is usually seen on the older leaves first, and then later in the season will move to the younger leaves as well. Brown spot, in Afrikaans called malroes, because I've heard it makes the farmers a little bit crazy. This one is sometimes earlier seen than the early blight, and it is smaller, rounder lesions. But these lesions also sometimes coalesce and then can blight the whole leaf. It's often first visible on the underside of the leaves, but this might just be because the inoculum is splashed up from the ground to the undersides of the leaves. So just to make it a little bit more confusing, we have some deficiencies that can also look like blight diseases. So manganese deficiency can sometimes be confused with a brown spot, and magnesium deficiency is sometimes confused with early blight. So be very sure of what you are seeing in your field before you try to control this. Then we also did fungicide sensitivity tests for potato South Africa, and we tested these 5 active ingredients. We managed to get a little bit larger population, at least, of the Alternarias, and we got them from 13 of our 16 potato production regions. So what we found here is that Alternaria, both our small and large spot, this means it's the brown spot and the early blight pathogens, from all the areas has a loss of sensitivity to QOI or the strobilurin fungicides. And then if you have a loss of sensitivity to azoxystrobin, there's also a loss of sensitivity to picoxystrobin, which means that none of the actives in that class will then be efficient against these isolates. Then all areas except for Sandveld and Ceres has a high prevalence of loss of sensitivity to the SDHI fungicide fluoparum, and this is especially in the early blight isolates. Then all regions has isolates with a loss of sensitivity to the dicarboxamide or pyrsomidone fungicide, and this is all both small sport and large sport. And then all areas also had a loss of sensitivity to tebuconazole. But the good news here, where there's a loss of sensitivity to tebuconazole, the difenoconazole still seem to be effective though. So this is called incomplete cross-resistance within a group. And then mutations that's associated with these fungicide sensitivity, loss of sensitivity has also been confirmed via PCR studies. So recommendations from FRAC, they say if there's a shift in your sensitivity, you have to remember your populations are heterogeneous. So you can turn this around. Not everything in a population will have a loss of sensitivity. If you want to avoid making this sensitivity shift permanent, you don't use continuous treatments with the same mode of action. Follow the recommended rate on the labels, and then your timing of your applications is also very important. And integrated disease management, of course. Do not only rely on the fungicides. So compiling a spray program, if you're looking at the recommendations from FRAC, they say do not exceed 1/3 of the number of applications with a product from the same FRAC group. So if you have 12 fungicide applications, only 4 may belong to the same group. Do not apply more than 4 products from QOI or SDHI. These are the 2 groups that have the most problems with a loss of sensitivity. And when you have mixtures, whether these are tank mixtures or co-formulations, do not apply more than half of your spraying program with one of these mixtures. And then if a loss of sensitivity in that area has been confirmed, only use those fungicides that has a loss of sensitivity in combination with others. So good news, the multi-site so far we did not find any loss of sensitivity. So our contact fungicides currently is still safe. So just to end off with, I just need to upset the apple cart a little bit quickly. Another blight disease that we are seeing more and more of in South Africa, I'm calling it emerging blight disease, is caused by Coleotricum coccoides, which is our black dot pathogen. So previously, this was seen only as like a post-harvest blemish on the potatoes, but more and more we are seeing this as a primary pathogen causing disease on green plants, on green living tissue, both on the stems and the leaves. These symptoms here, first off, if you look at it quickly, you might think early blight. We did not get any Alternaria from there. This was only Coleotricum coccoides. So please, if you see any of this in the field, we would appreciate more samples. We are still going to do pathogenicity trials to confirm Koch's postulates. And yeah, so if anyone has any interesting potato symptoms in the field, please contact us. We are always willing to look at these things and learn more. And together then we can innovate and evolve. Thank you.